Swayam Prabha. Digital India. Educated India.
Hi everyone, welcome again to the course of structural biology. Today we will start a new module which is the 11th module of the course, which is called protein engineering. But this is a very different module than what we had gone through, because whatever knowledge we have acquired throughout those previous modules, you could apply all of them in this module. I call the first class, which is an introductory class of protein engineering, a class with Mother Nature. We will see, is it actually a class with Mother Nature or not. Also, if you see on the right hand side, you will see that there is a script which is written in Bengali, which called Ha Ja Ba Ra La. It is a very famous writing written by novelist, storyteller, sci-fi writer Mr. Sukumar Rai. And if you guys do not know him, a reference will help you to understand who is Sukumar Rai. I hope you all heard about Satyajit Ray, the director, who got Oscar and who have created a lot of different films, including Pather Panchali. Sukumar Rai is his father. So, we will talk about this, why I have put it here, and let us start with protein engineering. Before going into the core of protein engineering, I will start at the topic which we generally talk when we start some landmark discoveries, like the wheel, the fire, the catapult and all those other ancient mechanical inventions which were nifty, but the cooking related innovation, the culinary innovation, bread, wine and cheese had at least as much impact on the world and altered human physiology in the process. Research, through investigation, chemists have announced that they identified the oldest actual piece of cheese, a 3,200-year-old leftover excavated from the tomb of Thames in Egypt. But there is reason to believe cheese goes back many millennia before that. Archaeologists have found 7,000-year-old pottery stainers that look as if they were used for cheese making. The making of cheese and yogurt can help explain why people took up herding some 10,000 years ago, even though adult humans' default state is lactose intolerance. So, this goes to the fact that this needs some thinking, and some clever and brave people realize that milk that spoiled is much easier to digest. We now know that what is actually happening. We know that microbes are digesting some of the lactose cheese making ushered dairy products into the human diet, and then for herding people, any lucky mutants endowed with lactose tolerance would have gotten more nutrition from all the dairy products. So, I talked about mutation. For today's discussion, especially for the module mutants, what we know now is variation of genes is very important. Genetic studies suggest that genes for lactose tolerance spread through herding people in Central Asia and the Middle East, and those lactose tolerant herder farmers replaced most of the indigenous hunter gatherers in Europe around 7,000 years ago. The first cheeses might have been paired with early wine. Scientists have scrapped wine and beer strains from 10,000 years old pottery. Genes that allow people to process alcohol started to spread in our primate ancestors, who moving from forest to savanna might have come across a lot of fallen naturally fermented fruits. Bread is even older. According to a study published July 2020, scientists found that they believe are breadcrumbs at a site that goes back 14,000 years, predating by 4,000 years the development of agriculture. Genes that improve the ability to digest starch seem to have spread among people whose diets included it. So, why I am talking about all those facts? Because today we are starting out our 11th module of the course, which is protein or enzyme engineering. It is a story of appreciating the beauty of Mother Nature is doing every moment, the mammoth task of maintain the living universe. In other words, it is about some creative geniuses who start thinking out of the box. So, in one side we are thinking, or we are trying to understand how Mother Nature is maintaining the entire living organism, the entire living world full of organisms, their all details. In the other hand, it is about some creative geniuses who is brave enough to think that we could mimic what nature had done, or we could have done better than that. This module will discuss about what, how, which of protein engineering. So, what is protein engineering? We will discuss. We will discuss how all those innovations we are talking about is actually happen on the level of enzyme, on the level of protein, and how we could alter those molecules, the functional molecule of life as I talked about. And then what is that? What is the changes? Then how we could be able to make those changes, and which protein to target, which application to target, and all those details. So, in definition, protein engineering is the improvement in the activity and usefulness of an existing protein or creation of a new protein function by making suitable changes in its amino acid sequence. As we have learned through the course that proteins are of two types, one is functional protein or enzyme. When this approach is used to modify the properties of any enzyme, then the specific branch is called enzyme engineering, which is a subset of protein engineering. Since enzymes are protein, enzyme engineering is a part of the larger activity of protein engineering. Protein engineering utilizes rDNA or recombinant DNA technology to introduce the desired changes in amino acid sequence of enzyme. In addition, increase in the level of production of protein is also part of protein engineering. Protein engineering is the process of developing useful or valuable proteins. It is a young discipline with much research taking place into the understanding of protein folding and recognition of protein design principles. So, this is very important, as I always told my young audience, that this is a very exciting time of biology, when biology is actually getting adulthood, understanding about the complex mechanism of how living organisms are doing all the transformation, and then creating something new. Let us say a chemical reaction where A to B is happening. Let us say this is not a chemical reaction, this is a biochemical reaction. So, there would be a enzyme. Understanding how the enzyme is doing the conversion from A to B, understanding which are the residues which take part in the substrate binding, in the catalysis, understanding which loop are flexible, and all those things help understanding the biochemical mechanism of the transformation of substrate A to product B. But now coming into the field of protein engineering, as I say, we are becoming adult, we are going for adventures. We are looking at, is it possible we could have take A1, which is a related substrate to A, A2, A3, A4 and A5, which are not supposed to be catalyzed by this enzyme, how we could make changes in the enzyme and make it possible the transformation of A1 to B1, A2 to B2, A3 to B3, A4 to B4 and A5 to B5. If this is a fundamental starting point of creation, we are not even restricted there. Now, we are even trying to take X and try to convert it to Y by the same enzyme where X is not related to A and Y is not related to B. And this is not only a very innovative idea, this is an idea on which our future is standing. Up to now, we have seen our synthetic chemists have done a lot of synthetic protocol where they make conversion of certain substrate to product. But as we all know that chemical reactions are generally taken care in an open vessel in a purified form where you could apply catalysts and different external things. They are expensive, they are multi-step. If you look at the properties of enzymes, they are mostly specific. Using their 3D structure, they mostly protect the substrate. So, their conversion are specific. What I mean? I mean, if you have a benzene ring and you want to transform a group into ortho, meta, para positions specifically, you need to first block the other positions, then do the reaction, then unblock it. Whereas, you could take the advantage of the enzymes which are providing you a nice 3D scaffold. It would be easily possible to much fewer steps, and as we all know that enzyme help you to shorten the activation barrier, you could have performed the reaction under normal temperature and pressure, removing or reducing a lot of cost. So, all those ideas are coming, but up to now we had understood, established the protocol and started establishing the fundamental principle of protein folding, which is the kind of starting point to go further. It is also a product and services market with an estimated value of $168 billion by 2017 and estimated as one of the most upcoming field in terms of finance. But before going into its application or details, let us start understanding from the basic what is protein engineering. Definitely there are two parts, one is protein, another is engineering. So, we all know throughout the last 10 modules, we have gathered a lot of knowledge about proteins. Proteins are large macromolecules that are comprised of one or more long chain of amino acid residues. We know that protein perform a vast array of functions within organisms, including catalyzing metabolic reactions, DNA replication, responding to stimuli, providing structure to cell and organisms, and transporting molecules from one location to another. Proteins differ from one another primarily in their sequence of amino acids, which is dictated by the nucleotide sequence of their genes and which usually results in protein folding into a specific 3D structure that determines its activity. We have also know the different level of protein structures. If you remember, I have always talked about the virtual experiment. If you close your eyes and I am giving you a bowl, so you could again perform that. So, close your eyes, do not open, then start imagining you have a bowl in your hand in front of you. The bowl is filled with 20 different color pearls. You have a string in your hand, you randomly pick up the 20 different color pearls and make a chain. This chain or a necklace of pearl, this is the primary structure. Then you get a good shape, that is secondary structure. Then you make a more perfect or particular shape, this is tertiary structure, and then you add other chains to make a beautiful necklace, that is a quaternary structure. So, basically primary structure is the amino acid residues, secondary structure is alpha helix and beta sheet, tertiary structure is combination of alpha helix beta sheet with the loops, and then quaternary structure is one monomer with different monomers forming oligomers. So, just to refresh, because these knowledge are very much important to understand where the changes are happening, what are the basics. But I will go quickly. Amino acids are building blocks. We all know that there is a amino group and there is a acid group, but there is more importantly a R group, and the chemistry of the R group is different 20 different amino acids side chains giving 20 natural amino acids, where basic amino acids are there, acidic amino acids are there, hydrophobic amino acids are there, polar amino acids are there. So, before continuing, if I ask you, do you think we could design above these 20 amino acids as building block? Yes. So, when we think about the field of protein engineering, the field of protein engineering is getting expanded to many reasons. One of the fundamental is the capability of introducing unnatural amino acids, the amino acids which are beyond these 20. We all know about the importance of peptide bonds, how the acid group of the first one and amino group of the second one, they polymerize, they do condensation by eliminating a water molecule and form a bond between the CN, which is called CONH or peptide bond. Very, very important, a peptide bond is a covalent linkage between two amino acids formed by the condensation reaction. This bond is formed between the amino and carboxyl group of two successive amino acids by hydrolysis of one water molecule. Secondary structure, as I talked about, secondary structure is alpha helix and beta pleated sheet. So, if you see alpha helix, they are formed by intra-chain into N plus 4th C double bond O NH hydrogen bonds. For beta pleated sheets, they form by again CONH, but they are face to face between two strands or in a parallel or in a anti-parallel. So, hydrogen bond between carbonyl and amine groups, and for beta sheets it is parallel and anti-parallel. Then tertiary structure, the tertiary structure refers to the overall spatial arrangement of atoms in a protein, stabilized by numerous weak interaction between amino acid side chains. Largely hydrophobic and polar interactions can be stabilized by disulfide bond, which is the only covalent bond formed after the protein comes in between the amino acid which is responsible is cysteine. Interacting amino acids are not necessarily next to each other in the primary sequence. Two major classes, fibrous and globular proteins. So, this is the example of fibrous one and this is an example of globular protein. Coming to quaternary structure, multi subunit proteins have another level of structural organization known as quaternary structure. A quaternary structure is formed by the assembly of individual polypeptides into a larger functional cluster. Quaternary structure refers to the number of subunits, their relative positions and contacts between the individual monomers. So, if you see, you have seen the globular protein myoglobin, here you see a multi subunit protein which is working similar to myoglobin. This is hemoglobin, these have 4 domains. In addition to that, more important is the presence of super secondary structures. Super secondary structures are structures which comes in between secondary and tertiary. They are part of a monomer, but the beauty of those part of that structure is if you see them, you know the function. So, super secondary structure directly announce the function, helping us to predict about the protein. When we talk about protein, if you remember, I always talk about there are few things which makes protein. This is called as the functional molecule of life, but in term of the computer to understand, if you remember my initial introductory module, I talked about, if you compare between carbohydrate, nucleosides like DNA, RNA, carbohydrate, lipid and all, you will see that they all have single bond in their joining, whereas the peptides apparently look single bond, but because of the lone pair in the nitrogen, they develop partial double bond. And as I repeatedly talked about, the presence of this partial double bond makes protein very, very unique from others, because of that there are restrictions in each unit helping us to develop proper models, because if there are two things connected by a stick, single stick, they could be developing millions of possibilities of rotation, which is not here in the protein because of the presence of the peptide bond, which is a partial double bond, the presence of other amino acids side chains like R1, R2 and all makes a lot of difference. So, that put individualism coming from the contribution of the side chain. So, you could include all those restrictions in the library, and that would help you to develop a model or help you to make basic rules which will help you to predict the structure of a protein. So, CN bond is there, in addition C alpha bond is there. So, C alpha is a psi angle and which is a dihedral angle, and C alpha n is phi angle. Then in the side chain there are chi angles. So, all those angles take very critical role in development of protein fold, but fortunately throughout our knowledge, throughout our availability of information, as I talked about the change of phase of biology with the innovation of sequencing, more importantly next generation sequencing, now we have millions of sequence and good amount of structure, but along with there is also improvement of computation. So, we could perform computer intensive simulations, we could apply algorithm of machine learning towards prediction, they are getting trained with the existing structure and sequence. So, all this is now helping us to apply or imply the basic rules of protein folding in designing a new one. And from this point I will talk about this in the next classes, but we are getting the courage to design something not at all thought by mother nature. I am talking about de novo protein designing. I will definitely spend a lot of time in the next classes, but you have to think about for a moment, oh my God, what is going on? De novo protein designing could give you a protein which is not present. What I mean, the fold at all is not present or not made by nature. With the establishment of top 7, a protein from David Baker's lab, we start believing that it is possible to design and actually express a protein, also make it functional, which is not at all included in the repertoire of our mother nature. We talked about Ramachandran plot, the plot of phi versus psi. Some combination of torsion angles are much more likely than others. The computed angles which are sterically allowed to fall on certain regions of plot, and it shows frequency of phi psi observed or residues in folded proteins. So, all those help us to develop the basic, and also energy distribution in different structural content according to flexibility presence in the bonds involved in the dihedral angles in protein backbone. Different level of rotation can be observed in proteins secondary structure contents. Depending on the rotation in protein secondary structure content, their stabilization energy also differs accordingly. So, for alpha helix, beta sheets, we have seen how they are different or how they are differently behave, helping us to again developing the basic rules of protein folding. So, this is all a refreshing memory of protein. Coming to engineering. Engineering we know is the use of scientific principle to design and build machines, structures, other items including bridges, tunnels, roads, vehicles and buildings. The discipline of engineering encompasses a broad range of more specialized fields of engineering, each with a more specific emphasis on particular areas of applied mathematics, applied science and types of application. So, together with what comes, I refer to that as the script you see called Ha Ja Ba Ra La. As I told, this is a story novel made by Sukumar Rai. So, I have taken the actual part in the Bengali script, but I have tried to translate it. So, the actual part is বেজায় গরম গাছতলায় দিব্যি ছায়ার মধ্যে চুপচাপ শুয়ে আছি তবু ঘেমে অস্থির ঘাসের উপর রুমালটা ছিল ঘামটা মোছার জন্য যেই তুলতে গেছি অমনি রুমালটা বলল ম্যাও কি বিপদ রুমাল ম্যাও করে কেন চেয়ে দেখি রুমালটা তো আর রুমাল নেই দিব্যি মোটা সোটা লাল টকটকে একটা বেড়াল গোপ ফুলিয়ে প্যাট প্যাট করে আমার দিকে তাকিয়ে আছে So, let us come to the translation made by me. I am first sorry, but I had to make the translation for my greater audience. It is extreme summer, lying leisurely under the pleasant shadow of a banyan tree, but still sweating heavily. My handkerchief was on the grassy land nearby. To take the sweat off, the moment I have tried to pick the handkerchief start roar like a cat, meow. What a problem! A handkerchief could shout was in mind. To my surprise, when I looked at that, it is not any more a handkerchief. It was transformed to a fat red cat and staring at me with a muscled moustache. So, do you believe that it could happen? Neither I. So, I exclaimed, I said, what a problem! It was a handkerchief and suddenly changed to cat? Impossible! The angry fat cat looked at me like he is representing Napoleon Bonaparte and said, impossible is a word which is nowhere in my dictionary. He continued, don't you see a egg converted to a quacky quacky duck? And what then? Then you don't think that how a egg converted to a duck? So, why you are surprised that a handkerchief is converted to a cat? At that moment, things started being clear to me. I start thinking, yes, there are so many things happen around us created by nature. Every moment how the whole living world is continued, how the transformations are made, how one cell become two cell, how everything inside the cell become doubled so smoothly, and I start thinking about the world. And also if you look at the universe, realizing the hugeness and remarkable capabilities of all living system, the amazing and breathtaking diversity of biochemical transformations they have innovated and continuously maintained with organized and stepwise manner. With the level of exact specificity and efficiency, all those systems are assembled from simple, abundant and renewable starting materials which are all around. How the character of parents are passing to the offspring, but with maintenance of their individuality. So, the offspring is individual. How? By making the shuffling of the DNA of mother and the father. Is not it something we are thinking when we actually start thinking about protein engineering? Things are getting clear to my mind thanks to the novel. So, just before I come back, I will tell you, if you follow the entire novel. It is really something which tell you a message around, but I have just picked. So, I will come to the story of evolution. You would also start understanding the fact that all the factors which are responsible for adaptation, optimization and innovation in the living world is protein, and majorly they are enzymes. You would also realize that Mother Nature maintains her own scientific group of protein engineers. They are working in a department called evolutionary office, and their function in one word is called evolution. So, their function, evolution executes a simple algorithm of diversification and natural selection, an algorithm that works at all level of complexity from single protein molecule to whole ecosystem. Isn't that amazing? Isn't that says you that if you could understand the basic principles how nature works, if you could bring the computation, if you develop the algorithms, if you run those algorithms correctly, you could also come up with designing of new ones or at least better ones. So, what is protein engineering? Protein engineering can be defined as any type of change or modification of the protein, and these will bring questions: what type of changes, how we could make those changes, is it possible to make those changes, why we need to perform those changes, what is the current scenario in the scientific community. I hope the continuing module would answer all of them, but another very important question is it not taking a step against the nature's creation? So, today I will talk about giving you a few overall idea and leads to next classes, but before that, as I used to do, I would definitely talk about the journey of the field of enzyme engineering or protein engineering. We say enzyme because most of the time it is enzyme. 10,000 BC, the Egyptians used fermentation for bread making and brewing, wine making, cheese making. So, that is the first use of enzyme without understanding. 1833, Anselm Payen discovered the first enzyme diastase, which is now also very important in the industry and called amylase. 1835, Jacob Bergilas coined the term protein. 1872, Maria Manasenia claimed the first evidence of cell-free alcoholic fermentation. 1878, Wilhelm Kuhn coined the term enzyme. He also started the field of enzymology by demonstrating cell-free fermentation. 1894, Emil Fischer lock and key hypothesis of enzyme. Takamine et al. Theory of enzyme synthesis used for separation and purification of enzyme. So, you might wonder why enzymes are coming over protein, because if you go back and think, as I always say, you start understanding, you start thinking, you will realize that in the earlier years in 1800 and early 90s, we do not have other technology, other instrumentation to detect a protein being folded or not. So, the only way to understand a protein is folded or not is to see its activity, because we know activity is on means the protein is folded, and that is why generally people used to target those proteins which have activity, mostly they are enzymes. I hope you understand that. 1897, Edward Buchner isolated zymes from yeast for fermenting sugar. In 1902, A. J. Brown proposed that enzyme reaction were initiated by a bond between enzyme and substrate. In 1903, Victor Henry proposed the fundamental equation of enzyme kinetics. In 1905, Sir Arthur Harden categorized zymogens into dialysable and non-dialyzable to help the protein separation and classification. In 1913, Michaelis and Menten kinetics, you all know the saturation kinetics, they demonstrated it with invertase enzyme and proposed the calculation of the rate of reaction. In 1918, Emden, Meyerhoff and Parmus proposed glycogen to lactate pathway, a part of the basic glycolysis follow up. 1923, Kimball and Marlin discovered peptide hormone glucagon. 1926, James B. Sumner isolated and crystallized the enzyme urease. 1929, John Northrop was the first one to isolate and crystallize pepsin. 1934, Wendell Stanley was isolated nucleoproteins responsible for tobacco mosaic virus activity. 1937, Hans Krebs and William Johnson established tricarboxylic acid cycle or TCA cycle. James B. Sumner crystallized enzyme catalase. 1944, D. Duvay purified the crystals of insulin. 1946, Sumner, Northrop and Stanley shared Nobel Prize for crystallizing proteins. 1948, Moore and Stein developed quantitative granular search chromatography method for fractionation of peptide, which is a very interesting and critical method for purification of peptide and use them in especially in the scale up. 1949, Friedkin and Lehninger proved the fact that NADP is the link between TCA cycle and ATP synthesis. So, both the factors, the TCA cycle and the ADP synthesis, both were identified, but the link was missing, which was done by them. In 1950, Moore and Stein developed the quantitative ion exchange chromatography for fractionation of peptides. Pierre Edman developed N-terminal sequencing method of protein, which is still valid. 1951, D. Duev identified the glucose 6 phosphatase as a target of insulin. 1952, Sanger sequenced insulin. 1953, Cunningham determined isoelectric point of trypsin. 1958, Daniel Khosland proposed induced feed model using conformational proofreading. 1962, John Kendrew and Max Peruz solved the first 3D structure of CIP hemoglobin using X-ray crystallography. Mortensen identified ferredoxin from Clostridia studied to understand electron transport. 1965, Monod, Wyman and Chengaks proposed concerted allosteric change in enzyme structure. 1967, Mitchell Moyle report chemi-osmotic hypothesis of oxidative phosphorylation. Spiegelman's experiment to understand the directed evolution of RNA molecule. 1970, Smith, Kelly and Wilcox isolate and characterize the first restriction enzyme. The type 2 was the first one to be isolated. 1971, so up to now I mostly talk about the understanding and identification of enzyme, protein related pathways, related model to calculate their rate and all the factors. 1971 is a landmark where Gutte and Merrifield designed the first modified enzyme bovine ribonuclease as DDT binder. 1973, Paul Boyer explained the mechanism of action for enzyme ADP synthesis. 1974, Claude, Deweb and Palade received the Nobel Prize for discovery of ribosome. You know, ribosome is very important because it is a big machinery which is involved in protein synthesis. Weibel and Pallad discover Weibel-Pallad bodies that store von Willebrand factor and p-selectin proteins. 1977 Stroud explained mechanism of zymogen activation. 1978 Arber, Nathans and Smith discover and characterize restriction endonuclease. 1978 Hall proposed the mechanism of directed evolution of enzyme. So, directed evolution of enzyme means how the nature is doing that, and this is another landmark towards understanding or performing evolution or enzyme engineering. 1985 Smith proposed for targeted mutation to a single protein. So, up to now it was theory, but now people are doing it. So, to do that you need to target and perform the change which we call mutation. So, Smith proposed for targeted mutation to a single protein. 1989 Richardson and Richardson proposed de novo design of protein sequences based on geometry and composition of similar protein. 1991 Francis Arnold, whom we will refer many time in our future classes of this module. Francis Arnold, we will talk, was awarded with Nobel Prize for her seminal contribution in directed evolution. So, this is the first breakthrough for her side. Francis Arnold proposed random mutagenesis to improve activity in Bacillus subtilis enzyme. So, random, up to now people know that you could do one change, you have to direct that, you have to make the process, but now they come up with something where random mutation is possible. Urey proposed design and production of elastin like protein, another contribution to protein design. 1998 Peter Kim developed computational design of multimeric non-natural right-handed coiled helices. I will talk about that in future. Stephen Mayo developed bitant and bound problem and energy function for solving protein design. 2000 Richardson published the penultimate library. This is a library of rotamers. So, when you start doing mathematics, doing calculation about a modification, suppose you have a serine, now you are putting a bigger one, tyrosine. So, what would be the apparent conformation? It is a very challenging and critical part, like how you say there would be a tyrosine, where the tyrosine would be actually you be there, what would be the exact rotamer. So, Richardson published a penultimate library which actually help you identify the optimized rotamer. 2001 Dunbrack published modified rotamer library with better functionality than what Richardson did. David Baker introduced Rosetta for ab initio modeling of proteins. So, I will talk about when I go to de novo designing, but the names and things are very important. David Baker is very important, like Francis Arnold. David Baker is one of the pillar of protein engineering, one of our scientists whom we are looking, and we hope that one day he will be awarded with Nobel Prize. He already have seminal contributions, many contributions already, which I will explain later. And this Rosetta, two packages, one is Rosetta, another is ITASR. These two have taken really important part in protein designing. Ab initio modeling, there are three type of modeling most majorly. One is knowledge based, another is energy based, and another is ab initio. ab initio is a modeling where the modeling is done from scratch. So, this is a very critical thing for protein designing. Ab initio I will talk about. 2002, Pierce and Winfrey proposed NP-Hurd model for protein designing. NP-Hurd is a mathematical model for polynomial, where in the proposal they consider a certain hardness or rigidity to the major body. So, that is the first model. 2003, Frank Russell performed directed evolution of function in beta barrel enzymes. David Baker identified novel fold using computational technique with atomic level accuracy. 2005, Stephen Mayo shows the effect of electrostatics on computational protein designing. Kingsford and Singh propose integer programming to choose optimal side chain conformations. 2007, Harbury introduced the concept of potential energy function in computational protein designing. 2008, David Baker performed computational enzyme design to engineer Kemp eliminase and retroaldolase. 2009, Maranas, another scientist who contributed a lot, Maranas and Khoury introduced switching cofactor specificity from NADPH to NADH of xylose reductase in yeast. If you see, the one of the biggest challenge in metabolic engineering is the specificity of different enzymes, one towards NADPH, another towards NADH. If you make those enzymes specific to one factor, that would be immensely helpful towards further designing. Koretme shows the effect of backbone flexibility on computational protein designing. Brooks developed charm force field. There are different force fields which we use when we simulate. I have already talked about amber, charm, they are very significant force field for proteins. 2010, Anderson created model of drug resistance using computational enzyme redesign. Jeff Gray introduced script based implementation of Rosetta modeling platform. 2015, Maranas and Pantages introduced iterative protein redesign and optimization suite of programs IPRO to redesign enzymes. We will talk about IPRO in little details. 2018, David Baker introduced beta barrel based porine protein design using de novo approach. 2018, Maranas and Chowdhury introduced software pore designer to precisely redesign bacterial channel pore size. Like you could narrow down, you could expand, you could make it specific. They had opened a huge area by this work. 2018, as I told, Francis Arnold received Nobel Prize for a contribution in directed evolution of enzymes. 2018, the same year, George Smith and Gregory Winter received Nobel Prize for phase display technique. So, when these two guys got Nobel Prize along with Arnold, a lot of people who know I am interested in enzyme engineering or protein engineering called me and told why these two, like a group who work on directed evolution and make new protein, and another people George Smith and Gregory Winter who make phase display, how they come together? Because when you are doing directed evolution, one of the biggest challenge is assay. You have to find the efficient ones, and the method of phase display is extremely critical in screening new variants created by directed evolution. That is the reason these guys awarded with Nobel Prize together. Another very important thing happened again, which I will talk about later. The first predicted model was developed, like the first predicted 3D model was developed by artificial intelligence or machine learning based tool, which is AlphaFold. They call it AlphaFold 2, which is considered, the performance is considered as comparable to experimental 3D structure. So, remember we start the course, we talked about there are so many sequences, and we have shown a graph that just in recent 2015 the number of structure was surpassed by number of sequence, and now they are way high because or thanks to next generation sequencing. So, now when we are getting millions or billions of sequences within few days, it is time to get their structures, and what better than prediction methods, because if prediction method shows comparable accuracy to experimental techniques, then you could make a lot of such predictions. You make available the structure of many proteins. So, in that point of view, this is extremely important. So, now we come to what, again to summarizing what is protein engineering. Protein engineering can be defined as any type of change or modification of the protein, and as I told, I am going to give answer of what type of changes, how we could make those changes, is it possible to make those changes, why we need perform those changes, and what is the current scenario in scientific community, with the question, is it not taking a step against the nature's creation. To explain all of these in short, because the 4th class of this module will explain them in details, I would talk about PCR. You all know a PCR reaction is a reaction which could amplify any type of DNA. So, what you need is a template DNA flanking primers, because DNA polymerase could not start from 0. So, you need flanking polymers for that to start. Then more importantly or most importantly, you need thermostable polymerase, which is TAC polymerase is one of the first one, but now there are PFU and many others, DNTP, the all DATP, DTTP, DCTP, DGTPs, and PCR buffer containing magnesium. All these you have to put in a thermocycler instrument, and you know that from template DNA you get exponential amplification, and in 30 cycle you would reach 2 trillion copies in theory. So, that says that with a very minute amount of DNA using PCR you could get a whole lot of DNA. You could vary PCR by varying temperature, cycle time and the temperature according to the primer, the buffer and the polymerase. So, why PCR was revolutionary, especially in terms of protein designing? Because first thing, what PCR is doing, PCR helps getting DNA amplified, right. So, this is a very big advantage, but in addition to that, suppose you have a gene and this part is a loop which is very flexible, you want to see that how the protein behave without this. So, what you do, you just take that out, which is called deletion mutation. Similarly, you could perform insertion mutation, you could put anything, you could also do. So, these are big mutations, you could do point mutation. Nowadays, there are, so here also you could do side directed, you could do random, you have error prone PCR, all those whatever you thought now comes to reality thanks to Kary Mullis and its innovation on PCR. Innovation of PCR make landmark towards protein designing. Again, I am not going into details, you know the concept of DNTP. If these guys are not there, then you get something like that, and these added with PCR amplification, another good part, lead to Sanger sequencing, where you could read, and you could not only read, you could read 800 nucleotide base lengths, the accuracy is 100% and it is considered as gold standard. So, you make mutations, you make amplifications using PCR, not forget you make cloning and everything related to, but with these you start reading. So, why Sanger sequencing was revolutionary? Because one, it helps reading genes, helps reading mutations. So, you have performed mutation, you have to check the mutations are done or not, that is done by Sanger. Still now it is the gold standard, and then it helps towards reading loads of important proteins. Then it goes to genome sequencing, from there transcriptome sequencing, metagenomes, and the list is endless. More and more introduction of technique is coming, more and more it is helping you to read, like let us say chip, it helps, chip based sequencing is helping to understand where the transcription factors, majorly factors binding, helping to understand the regulation. So, when you are doing protein engineering, enzyme engineering, it is not about the DNA, it is also, so it is a DNA or gene, but it is also about gene regulation. That is beautifully done because of innovation of Sanger sequencing and further techniques. So, one question every time comes in people's mind, is it not taking a step against the nature's creation? The answer is not at all. And what is the proof? The proof is, today we learn things, today we learn words, today we say we are doing protein engineering, so a group of people come, no, do not do protein engineering, do not play with nature. But think about, for thousands of years human have been making cheese, wine, bread, so they are selecting the best makers, the yeast, the bacteria to make yogurt, the lactobacilluses. Also, we are performing breeding of animals and plants through the selection of organisms with desired properties, which was also scientifically documented by Gregor Johann Mendel. So, for most of this time, without even knowing they were doing it, humans evolved and optimized enzyme and binding protein over many generations, and nothing happened. Now, what we are doing, we are just gathering those information, we are just taking them and trying to shift into the direction in a more organized way. That is the only difference. So, coming to the summary, so now we know whatever you want to do on the level of protein or enzyme, you have to go back to the DNA. So, what could be protein engineering? Let us start from very simple thing. You have an enzyme and you are trying to overexpress the protein. It is not overexpressing. Now, you model the protein and you find few areas, few loops which are not connected to the rigid body, the main structure of the protein. You cut them in the genetic level and you clone it, you get higher expression. So, that is a basic protein engineering we do regularly in the lab. Definitely what we do, we take the original protein and we compare the activity with the recombinant protein. So, that is the basic one. Then stability. Nowadays, there are a lot of concern about stability because we are not happy with the production of the enzyme, let us say food. With the huge enhancement in the population, we need more food, we need more generation of anything. So, we use enzyme and we want to use the enzyme into industry. For that, enzyme have to be stable, thermal stability, stability with pH, stability with solvent, denaturants and many other. Also, we want to reuse the protein. For that, enzyme immobilization is a great thing. So, how we could modify a protein so that we could, or enzyme we could immobilize it, that is also protein engineering. Then from there to go for changes as I explained through big mutations and point mutations, all are possible. Then we could also do random mutagenesis. Here, as I told, the challenge is screen the assay system. Then we change what we call blob change, or suppose this is a protein which form a tetramer because of this unit. So, we cut it and this is protein A, we use in protein B this one, so that this helps making a tetramer of B. So, that is what fusion proteins we say. One protein have one domain which is good for reaction 1 and the second domain is non-catalytic. We replace it with another domain which do perform reaction 2, also engineer. And last but not the least, it is about de novo, a design of a enzyme which have no existence in terms of fold in the nature. Now, you see I have kept this place blank, there is a purpose. So, if this is the summary, I have a dream for performing abiotic reactions in the laboratory which would change the spectrum of the world. What I mean? Let us say I have a cytochrome P450. By the way, you will see cytochrome P450 engineering if you go to Francis Arnold's work. I will talk about that too. They had done a lot of work there. As I told, my group also work on protein engineering, and one of the work we are doing cytochrome P450. So, we are making definitely mutations towards function, but if you ask me what is my dream to work here, this is about you have cytochrome P450 where there is a porphyrin ring binds to iron, and we know that this enzyme is a oxygenase. So, it perform CH oxidation which is to introduce CH functionality, which is a dream of many synthetic chemists. So, let us say if we replace iron with other similar metals and set up reactions, oxygenation reaction, which generally perform in multi-step with expensive metal catalysts in abiotic condition, we could have not only lower the steps, but also makes it extremely cheap, and these processes would really contribute towards development of many new compounds. So, this is something you could think about. We will talk about all those, what are the process and all. So, coming to the end of the class, in the early days of biotechnology, the biocatalytic reaction was adjusted to meet the optimal condition of the enzyme used. We always try to understand the enzyme, we check the enzyme's condition, like how it works in which temperature, which pH, which salt concentration and all, we call it optimization. Now, we will still do that, they are basics, but nowadays we are able to engineer the enzymes themselves. Via this method called protein engineering, the enzymes are matched to the target substrate or to the reaction conditions. Furthermore, protein engineering can even introduce alternative reaction mechanisms for a given enzyme, as I told earlier. There are two complementary approaches, like all those processes comes under two major branch: rational protein design and directed evolution. For rational design, a large set of enzymatic parameters are needed, but only a few hotspots in the sequence are mutated, which keeps the screening effort low, because you are doing it knowingly. It is called logical design or rational design. Another one is directed evolution. Directed evolution contains only basic information about the protein, because the gene sequence is mutated randomly mostly, but the screening effort is very high. How we do that, how we perform that, all of them we will be see in the following classes. With this, I am making an end to this class. Thank you very much for listening. Thanks.